Good morning or good afternoon, everyone, and welcome to Genome Webinars. I'm Julia Caro, Managing Editor at GenomeWeb, and I'll be your moderator today. The title of today's webinar is Using a Mass Spectrometry-Based Platform to Perform Population-Based Newborn Screening for Cystic Fibrosis in the State of Illinois. This webinar is sponsored by Agena Bioscience. Our speakers today are Dr. Vineet Dhiman, Clinical Laboratory Scientist at the Illinois Department of Public Health, and Dr. Robin Ebbets, Senior Manager of Global Pharmacogenetics and Clinical Genetics in the Department of Scientific Affairs at Agena Bioscience. You may type in a question at any time during this webinar, and you can do this through the Q&A panel, which appears on the right side of the webinar presentation. We will ask our speakers your questions after the presentations have concluded. Also, if you look at the bottom tray of your window, you will find a series of widgets that can enhance your webinar experience. So with that, let me turn it over to Dr. Dhiman. Please go ahead.
Right, thank you, Julia, for that introduction. First off, I would like to thank GenomeWeb for hosting this webinar and Agena Biosciences for giving me the opportunity to talk about this subject. So again, my name is Vineet Dhiman, and I'm a scientist with the Illinois Department of Public Health. And today I'll be talking to you about our experience using the Agena MassARRAY System, specifically with cystic fibrosis newborn screening. So first, I'll give a little background on IDPH, a little bit about our health department, and then talk about some of the interesting caveats of the assay as it pertains to newborn screening. So next slide. So IDPH, just to give you a brief overview, organized in 1877 as one of the state's oldest agencies. We have headquarters in both Springfield and Chicago cities in the state of Illinois, which includes many regional offices. We have 3 state laboratories across the state with over 1,000 employees. IDPH oversees a plethora of different programs that affect the health and well-being of every Illinois resident and visitor. These are included but not limited to food, water, drug testing, hospital, nursing home licensure, vital records, emergency preparedness, and of course, newborn screening. So the goal of newborn screening is for early detection of conditions which we have early and timely interventions and can lead to the elimination or reduction of associated mortality, morbidity, and disabilities. So in the US and internationally as well, whenever there's a new baby that's born, typically what happens is that there's a heel prick that's involved, and the blood is submitted to various laboratories for screening of various disorders in hopes to catch some of these disorders earlier before any clinical presentation, in the hopes of being able to treat those disorders and to provide better quality of life for those prospective babies. So just some general requirements of newborn screening. The condition should be important to public health. There should be facilities for both diagnosis and treatment for each of the conditions. There should be a test or examination that's available for that condition. The natural history of the disease should be pretty well understood and well cited in the literature. The cost of finding a case should both be economically balanced in relation to the medical expenditure required, and the case finding should be a continuous follow-up process after screening of the children and babies are conducted. So to give a brief overview of the different disorders that we've introduced at the Illinois Department of Public Health over the past, in this case, 50, 60 years, starting with PKUs in the mid-60s, and you can kind of see this slide, congenital hyperthyroidism in late 1970s, amino organic and fatty acid oxidation disorders in the early 2000s. CF, we started screening for CF in 2008, and more recently we added adrenoleukodystrophy, as well as spinal muscular atrophy as our disorders that we screened for in 2019 and 2020. So a general overview of the newborn screening timeline. At day 0, you have your birth of the baby. Typically, the blood of the baby is collected post 24 hours usually via heel prick, and then the blood is spotted on specific cards, which we call U3 cards, which I'll discuss in a minute. The specimens are then transported to the laboratory where we conduct the testing, and depending on the test, whether the specimen will be negative or positive for a specific disorder, reporting the results typically takes about 5 to 15 days, depending, again, on whether there's confirmation testing that needs to be conducted. Each disorder has a different test that can take some time. Sometimes the controls for a specific assay may not work very well, so there's a bunch of repeat testing that has to be conducted. So there's a lot, to your typical laboratory nuances as far as testing is concerned, that can happen. But we do try to get results out within 2 weeks because obviously with all of these disorders, time is of the essence. Some disorders more so than others where you want to get treatment done as early as you possibly can. So this is an example of the Illinois newborn screening specimen card where you have the demographic information that is required for each baby, and baby, the name, first name, last name, the mother's information, the gender, nationality, and then some important things like birth weight, whether it's a meconium ileus was present, and then the provider information as well. And on the right side of the card, as you can see, this is where the blood spots, where the blood from the heel prick is spotted on the card in these circles. And essentially what we want is complete saturation of the blood on each one of these circles, so we are able to test it properly in the laboratory. This is another example where, again, you have the heel prick on the foot, and then this is an example of a properly saturated card where you can see each circle, you see proper saturation, proper spotting of the blood on each of the circles indicated. Sometimes you get a lot of smearing if the heel prick is pretty messy. Sometimes what you'll see is that the blood isn't saturated through the card. And this, the Guthrie card is kind of reminiscent of maybe like Whatman paper type of material a little bit, just to kind of give you an idea of what it feels like. Right. So as I kind of alluded to a little bit earlier, there are times where there will be unsatisfactory collections. So these are include but not limited to, again, no or insufficient blood that's collected, a contamination with foreign substance. The sample is not dried properly, usually it's about 4 hours after spotting the blood on the card, layered or clotted collections, the specimen's heat fixed, there's a laboratory accident, or it's even not on the approved card. So typically, if we do have an unsatisfactory collection or unsatisfactory card, we will go back and contact the provider to request another submission from that specific baby. So each specimen card received is punched at least 12 times. So we perform a 3.5 millimeter punch. And so each card is punched a minimum 12 times. And these are the examples, these are for, and the slide shows the examples for each test that's being tested. So 2 for the lysosomal storage disorders, which include a variety of disorders, 2 for galactosemia, 1 for SCID, SCID and SMA, 1 for congenital hyperthyroidism, and so on and so forth. And then what we'll be focusing on today is the cystic fibrosis arm of our testing, where there will be 1 spot punched for IRT, immune-reactive trypsinogen, and then if that's positive, 1 punch will be then be punched for cystic fibrosis mutation analysis. And I'll get a little bit into this in a few slides. All right, so a little bit of background on cystic fibrosis. It's the most common life-threatening autosomal recessive disease in the US, incidence rate about 1 in 4,000. It's an inherited disorder that causes severe damage to the lungs, digestive system, and other organs. And it primarily affects cells that produce mucus, sweat, and digestive juices. So cystic fibrosis is caused by mutations in the CF transmembrane conductance regulatory gene, as it will be known as CFTR going forward. This first gene was first sequenced fully in 1989. It's a very large gene, 250 kilobases, 27 exons. And to date, there are more than 2,000 mutations that have been identified within this gene. And you could go to the CFTR2 database, just Google CFTR2, and it kind of gives you a full profile of all the mutations known. However, only about 10 to 15% of those mutations have been confirmed to be disease-causing. So about 200 or so. And those 200 or so mutations are known to occur, differentially across different races, different allele frequencies, so on and so forth. Excuse me. So the defective gene, so if there is a mutation and it causes a defection in the CFTR gene, this causes various fluids to become sticky and thick. And instead of acting as lubricants, these fluids essentially plug up ducts and passageways. And this occurs especially in the lungs and pancreas. And it causes a whole source of issues. Again, digestive, breathing, so on and so forth. The earliest clinical presentation of cystic fibrosis occurs with meconium ileus at birth, which is in about 20% of the newborns. And so what's happening is that the first stool that a newborn baby has, instead of coming through nice and liquidy, it is a bit more thick. And so it basically blocks the small intestine of the baby, and then you'll get swelling of the baby's internal organs, especially the small intestine. All right. So the traditional gold standard clinical test is a chloride sweat test. And this is typically performed at the hospital or clinic anytime after 48 hours after birth. And typically what you're measuring is the chloride levels, the sweat chloride levels in these babies. And any baby with levels above 60 millimolar per liter is typically seen as a kind of as positive. Unfortunately, this is not always clear cut, and there can be sometimes you might not collect enough sweat from the baby where you'll get quality not sufficient. You might not detect the sweat properly at the appropriate level. So there are issues with this diagnostic testing. And so enter newborn screening for CF. So newborn screening for this disorder first started in 1985 in the US, in Colorado, and then it was fully implemented nationally in all 50 states by 2010. And this, there was a study in 2005 in the journal published in the Journal of Pediatrics that compared babies where they were diagnosed or screened for CF via newborn screening versus traditional methods. And you can see by both the height and weight of the babies indicated in black lines are overall higher in babies screened by CF, screened by newborn screening for CF as compared to the traditional clinical presentation methods. So obviously with CF, with newborn screening, you are getting the earlier detection. And again, earlier detection means earlier treatment. And so how do we screen for CF? So IDPH, in addition to many states across the United States, utilizes an IRT DNA CF screening algorithm. This is a two-tier test where the first screen, we're looking for the levels of immunoreactive trypsinogen. So this IRT is produced in the pancreas as trypsinogen, which is the inactive precursor to trypsin, a serine protease that cuts peptide chains, essentially breaking down food for digestion. So in patients with CF, the thick mucus actually blocks the pancreatic ducts. So this prevents trypsinogen from actually reaching the small intestine to be converted into trypsin. And so as a result, you're getting inefficient breakdown of food, as well as essentially blockage of various fluids in these various organs. And so IRT is detectable in blood. And so that's why, with the blood spotting from these, from these babies, we're able to look for the IRT levels. And newborns with CF may have elevated IRT. Unfortunately, just looking for IRT is not definitive. It can produce a lot of false positives because it can be inaccurate if, for example, the blood is collected less than 24 hours post-birth. And that's because during the birthing process, there can be slight damage to the pancreas to the baby, which creates this same elevated IRT levels. And so whenever we see this in the laboratory, any baby who's whose blood was collected below 24 hours, we typically rule that as unsatisfactory for cystic fibrosis, for IRT screening for cystic fibrosis, because of the fact that the IRT levels just aren't reliable. And so, however, for all other babies that we screen post 24 hours, any newborn with elevated IRT, and by elevated IRT, we typically look for any IRT levels that fall in the top 4% of values for that given day, is automatically reflexed for cfDNA mutation screening. And so this is an example of the kind of your traditional IRT DNA screening algorithm for CF screening, where the newborn blood spot is collected and sent to the laboratory for testing. If it falls below the 97th percentile or 96th, each state has slightly different criteria for what they use as IRT positive. So if it's not within the top 4%, at least for IDPH, we report that as a normal for CF screen. However, if it's in the top 4%, a IRT repeat or confirmatory is conducted. And if that repeat is still elevated, it is sent for CFTR mutation analysis, which you can see over here. However, if that repeat is normal, or if it just doesn't fall within the top 4%, that screen will result as normal. So as with CFTR mutation analysis, there are a number of different results that can happen. We could have 0 mutations, 1 mutation, or 2 mutations screened. So with the 0 mutation case, what will happen is if it's 0 mutations and the IRT level is below 150 nanograms per mil, or in IDPH's case, we use a limit of 170, that indicates low risk for CF, and we result that as a CF normal. However, if we do not see a mutation, but we see high levels of IRT above 170, we will still report that out as a CF positive screen, just no mutations, and it falls into the category of very high IRT. And then obviously, if we do detect a mutation, whether it's 1 or 2 mutations, regardless of whether it's 1 or 2, we then repeat that for confirmatory testing. And then if we do see that again upon confirmatory testing, we'll result that out as screen positive. And then we will notify the hospital or the pediatrician, whoever is essentially responsible for that baby, the health club provider, for them to then conduct their diagnostic testing and essentially, refer to a genetic counselor if needed. And so everything kind of in the green, it happens in the laboratory, and everything here in the red, the sweat test, that happens in either the hospital or the clinic for where the diagnostic testing typically occurs. So as mentioned previously, there's over 2,000 known mutations with only about 10% disease causing. The most common mutation that's observed is delta 508, which is observed in about 70% of alleles. And so when CF DNA screening was kind of created and became something that was possible in all of the states, the American College of Medical Genetics recommends that at least 23 of the most common CF mutations should be screened for CF. And that's indicated in yellow in this image here. All these mutations, these various mutations you see, are the ones that are essentially included on every CF screening panel for newborn screening. And then the other 51 here in this accompanying image are the ones that Agena offers for their off-the-shelf CF assay. Depending on the company, some of these mutations might differ slightly, and the reason for each company choosing these mutations might differ slightly depending on if they're offering their assay for a specific population, because depending on your ethnicity or race, you might be just predisposed to some mutations more than others. So obviously you just want to kind of pick the best panel that kind of encompasses everything that you might see in your population. So how do we actually set up this assay at IDPH? So we try to make everything automated. We want to make sure we remove as much human error as possible. We don't want the samples to become mixed up in the 96 or 384 well plates that we use to do the PCR amplification, or where we punch the blood spots in. And so we have these EP Motion 5075s by Eppendorf, which is an automated liquid handler, and they perform all the PCR, methionine alkaline phosphatase treatment as well as extension. And again, it just kind of erases the human error part that could go wrong in any laboratory experiment. And then on the screenshot below, these are our two Agena MassARRAY systems that we have. They are two instruments. They can run 4 plates between them, 29 specimens each at a time. And so what do I mean by that? So this is an example on the left of the DBS punch plate that we do when we punch for cystic fibrosis. We have 2 positive controls, positive control A and B, and this typically screens for, these positive controls typically are about 25 or so of the positive mutations across the panel that we are ensuring are amplifying properly. We also have a negative control. And then we punch 29 different specimens on the plate. And so the way that the Agena cystic fibrosis assay is set up is that in order to test the 74 mutations on their panel, they break that up into 3 different PCR reactions. And so what we end up doing is that we essentially are speccing each of the 29 specimens 3 times on 196 well plate. And so as you can see, with the colors indicated here, we have PCR amplification A, PCR amplification B in blue, and PCR amplification C in green. And this will allow us to essentially assay for all 74 mutations across each specimen. So after we get everything onto the Agena instruments, the assay is run. This is an example and screenshot of the analytical software that we are presented with for us to do our analysis to look at the results. So in the top left corner is the assay ID, and this could be broken up in date, assay type, and the way the customer, i.e. yourself, wants to organize the data. There's a field for each of the different assays that you're selecting. This box here contains every single mutation on the panel. This is a snapshot of your 96 well plate, where the different colors indicate how efficient or the quality of the results in a given well, where green typically means that every mutation works. Red means that it's usually a failure or there's no mutations that were actually detected. And then you could have different varying colors from dark green to light green to yellow, which is basically a spectrum of how well that, of how efficient that specific well on your plate amplified. So this here is a plot that indicates what each sample is presenting as, heterozygous wild type, homozygous wild type, homozygous mutant, or heterozygous mutant. I'll get into the different colors in a bit. This is your spectrum of your peaks, where it looks for your wild type peak, the mutation peak for each specific mutation. And then this panel here essentially indicates every single mutation for a given well. So zooming in a little bit more on the plot, this provides a really good snapshot of all of the specimens tested for a given mutation. So you can see that, for example, the mutation S549N. You'll see that the yellow squares here, the green squares here indicate heterozygous mutation, and they typically fall between the 2 dotted lines on the outside. The yellow triangles here indicate a homozygous mutation. The red circles indicate a failed detection or a failed amplification, basically a failure, which usually begs a repeat for that specimen. And then the majority of your specimens will be wild type, which is indicated here as blue triangles. So for the most part, the assay works really well. The specimens will either appear as wild type or either have a heterozygous or homozygous mutation. There are some cases and caveats to data interpretation that I'm going to go into that you should be aware of when considering this assay for your own means, whether it's newborn screening or something else. So for the first caveat is basically data interpretation, which is exemplified with the delta 508 mutation. So specifically with a delta 508, there are different mutations that could happen at the 508 position. With this being one of the most common mutations, usually what you'll see is with the green squares here, you'll see a heterozygous. This usually indicates a heterozygous mutation. However, special attention must be applied to anything that's homozygous for the delta 508, because you also need to see what is the status of the F50C in that same patient. So, for example, despite there being a homozygous mutation for delta 508 deletion, this patient is also presenting with a F508C homozygous mutation. Essentially, what's missing is your wild type allele, right? And because there is no wild type allele present in this baby, you have one allele with F508C and one allele with the delta 508 deletion. And so the end result for this patient would be a delta 508 heterozygous mutation. This is in contrast to this observation where in conjunction with this homozygous mutation of delta 508 deletion, you get an amplification failure of the F508C in this same baby. And so in this case, the end result would be a delta 508 homozygous mutation. And so we actually had an issue with this specific mutation. Because F508C is a benign mutation, it's not disease-causing, we had actually initially hidden the results of this specific mutation from the Agena view because we like to try to keep the analysis portion as simplified as possible for our technicians. So keep in mind that our technicians don't have PhDs. They're typically with the bachelor degrees. They don't have specialized training, and so we're trying to keep this, I guess, quote unquote as dummy proof as possible. However, because we had to ignore the 508C mutation at the time, we had mistakenly called out something as homozygous 508 homozygous deletion. However, going back to the data, when we unblinded the F508C, we noticed that that wasn't the case. This one actually did have F508C present, and so instead of a homozygous, we had to fix that call out to make sure and correct it as a heterozygous mutation. And so another caveat to interpretation is amplification failures, where, as you can see in this plot here, you have a bunch of just red circles all over the place. This is typically seen when your PCR amplification does not work at all for a given mutation. And essentially what happens is that you just have to repeat that specific specimen because there's a mutation that failed. And this happens from time to time. So there are periods of time where it doesn't happen at all. And as with all science, sometimes it'll just magically happen, and for whatever reason, you can't figure out why. And no matter what you do, it's like, why is it not going away? Usually what we'll end up doing is that we'll try to kind of replace all the reagents fresh. We will try to clean everything with a DNA off type of reagent. And more often than not, that will kind of fix a lot of these amplification failures. And the last caveat to interpretation, it's a bit more of a difficult to deal with, is these low signal mutation callings. And so what sometimes will happen is that with some mutations, for example, this R1158X that you see here, you'll see that it's basically red across the board, right? There's a few areas where you'll see the blue triangles where the interpretive software was able to call it a wild type mutation. I'm sorry, a wild type call for some of the specimens. However, the vast majority are red circles. However, and you can see one of these red circles is highlighted where you can see a peak that's present here, right? Under the wild type peak. However, there is no mutation peak. However, despite a weak peak being present, the interpretive software is not calling it, and that's because it's not crossing the minimum threshold that the interpretive software requires for it to call. And what we found with some of these mutations is that regardless of how many times we repeat that specific specimen, we will consistently get these weak peaks. It's a clear peak, but it's just too weak for the baseline interpretive software to call. And so what we'll have to do is that we'll have to manually look at every single specimen, make sure that the peak is present, and manually call them as wild type or heterozygous mutant or homozygous mutant, whatever we need to designate it at that time. And so this is where, in some cases, you'll have, as much as you want to automate the entire process, there is some manual interpretation that is required for some of these mutations. And obviously, this is this could be because some of the DNA blood spots that we receive isn't totally pure. The extraction may not have worked as ideally. In a perfect, even though ideally, when you're using pure DNA, these mutations light up perfectly. However, when you're dealing with real clinical specimens, you're not always going to get that perfect specimen. And so sometimes you'll have to deal with less than perfect conditions in which you're making some of these calls. And so just to kind of give a brief finale of the volume that we do here at Illinois Department of Public Health. So we routinely screen about 155 to 165 specimens each year in our newborn screening program. For cystic fibrosis specifically, that number is about 6,200. So we have a pretty high volume. Illinois is, I believe, is the fourth most populous state, having one of the biggest cities, Chicago. So we do have a large population that we have to account for. For example, in the year of 2020, we screened 6,160 specimens. Of those 6,000 or so specimens, we reported out 542 screens. 23 were ultimately diagnosed as CF with cystic fibrosis. 343 were diagnosed as carriers, where we only determined one mutation, and after diagnostic testing, they were negative for CF in the clinic. 107 were diagnosed as normal, and all of these were essentially our high IRT callouts, which essentially could be false positives. And then we still have 69 pending cases. And this goes into the fact that follow-up with each one of these cases is a huge effort unto itself. And not all providers always get back to us with the diagnostic information of each case. And so our follow-up team here in Illinois does a fantastic job, just following up with all disorders that we call out positive for. And, we try to make sure all the data is complete for every positive screen that we do call out. So keep in mind, we are a newborn screening. We're performing newborn screening here at IDPH. It's not diagnostic testing, right? It's a screen. So when we report out these positive screens to the hospitals providers, it's still unto them to do that diagnostic follow-up. We're just trying to catch the most, we're the first step in trying to catch these disorders as early as possible, basically days after these babies are born. So when we compare the Agena CF assay to what we were previously running, we found that we actually called out about 100 more cases, more positive screens with the Agena kit than with our previous kit, which only did about if I remember correctly, it only did the minimal 23 mutations. So that's the power in this Agena kit or any kit where you are able to screen for more mutations. You have more, you're able to catch more and more of these cases, and hopefully more of the positives as well. consistent with what's published in the literature, the most common mutation we did observe in our data set of every year since we've started this is the F508 deletion. Some of the common mutations that we've seen in our population include W1089X, R1066C, and the 2055 deletion 9 to A mutation. And so I guess the one caveat with the Agena is that it's a great assay. It's a great assay as far as being able to automate it. It's very it does well being able to set up for automation. There is a high barrier for data interpretation because it's not an automated process. There is manual interpretations that need to be made. So there is that training for your technicians, your bench strip technicians, for them to, when they're reviewing the data. At IDPH, after technician review, each batch, every specimen undergoes 2 supervisor reviews just to ensure everything is being called out properly before we officially report out any positive screen, or positive or negative. So there is that that caveat as well, in that if you are looking for a complete automation from start to finish, while the bench part of it may be easy to automate, there is that caveat with data interpretation where not everything might be automated. And with that, that concludes my portion of the talk. And next, I'll be handing it over to Dr. Robert Everts for him to discuss more so on the Agena side. Thank you.
Well, thank you, Dr. Dhiman, for this very informative presentation. I assume you can all hear me right now. And I will represent here Agena Biosciences, and I will talk a little bit about our company and about the workflow and platform, as well as the CF offerings we have. So as I said, I'm Robin Everts. I'm a senior manager in clinical genetics here at Agena Bioscience, and I work in the Department of Scientific Affairs. So let's get started. So Agena Bioscience is a worldwide company, and we are providing targeted genetic analysis. So we actually do genotyping, and our focus is on timely, accurate, and targeted results that help you decide in your laboratory what to do with a sample or not. And we are actually located worldwide in different places. Our headquarters are in San Diego, and our headquarters are in San Diego. And then we have 3 offices in Hamburg, Shanghai, and Brisbane. So as Dr. Dhiman already talked about, there are thousands of mutations in CF, and there are, of course, different varieties. They are classified into class 1 to 5, and sometimes classified in 1 to 6. And class 1, of course, is a class where there's more severe symptoms, whereas with class 5 or class 6, the symptoms are not as severe, or they happen later in life. And as Dr. Dhiman told about, like Delta F508 is the most prevalent mutation that is out there. As Dr. Dhiman also was talking about, the ACOG or ACMG recommends 23 mutations. And when you look at those mutations, they do really well for people from Caucasian descent, with about 98% of carriers finding. However, in other ethnicities, such as Asians, you might only find about 49% of carriers or people affected by CF. So a lot of people actually will, in the future, look for other mutations also. And that's something with our IPLEX Pro CFTR 74 mutations, Dr. Dhiman has shown that it actually finds more affected people and more carriers than the standard 23 ACOG set. So when you're looking at a platform, there are different buying considerations, of course. Open versus closed, ease of deployment. Do you have to do a validation or not? What's the workflow? How many personnel do you need for it? Economics, what's the running cost? Is the reimbursement? And then, of course, coverage. As I talked about, there's the ACOG ACMD 23 CFTR mutation. You can also do whole genome sequencing, where you find everything under the sun, including a lot of unknown variants of unknown significance. Or you can do something in between. And that is where Agena comes into play, because we can actually cover from, say, 23 assays up to a couple of hundred assays. And Dr. Dhiman just discussed the fact that only about 10 to 15% of the 2,000, so only about 200 to 300 of the assays or the mutations actually are known to be CF causing. So how does our platform work? So we have a very basic workflow. We start out with a PCR reaction. We have a PCR where we amplify the alleles of a specific mutation. So in this case, you have an A allele and a G allele for a particular mutation. We amplify fragments about 100 to 200 base pair in length, and we amplify about 30 different SNPs into one reaction. So one well on a 96 well plate can contain up to about 30 different SNPs for a particular sample. Then after the PCR is done, we do a shrimp alkaline phosphatase treatment. We dephosphorylate the unincorporated DNTPs. So the DNTPs that are left over after PCR cannot be used anymore. And after that step, we add a extension primer cocktail, which has an extension primer that lies next to the SNP of interest. In this case, you can see that here in the fourth pane out here. You can see that the primer will actually incorporate an A here or a G here. And one of the nice things of our technology is that because we add all the 4 different terminators, A, C, G, and T, even if an assay is triallelic or quadrillelic, we actually will incorporate the right base on the right position. And we don't have to worry about missing like one of the alleles because we are limited in like how we are running this. Once this reaction is done, the IPLEX extension reaction, once that is done, we desolve the reaction. It's still all in a 96 well or a 384 well plate. We dissolve it, we add water, we add resin, and then we dispense that product onto a spectral chip, and that is loaded into the MassARRAY, and then the MassARRAY will actually analyze the data. It will create peaks for all the different products out there, and in this case, these 2 peaks are the A and G alleles of this particular assay, and they will be reported as A and G. So getting a little bit more into detail on how this works is this slide here. So how do we know that that A and G peak were actually the A and G for the particular assay? So what we do is we have a single base extension primer that lies next to the SNP of interest, and it incorporates either a T, a G, an A, or a C here. And so based on the incorporation of these dideoxy terminators, we can actually predict upfront already what the actual masses are. Like the C allele is going to be 5,971 Daltons, the A allele is going to be 5,995 Daltons, etc. If we then run this on the MassARRAY, what you'll see here is that the primer itself on the left side here was about 5,725 Daltons, but by incorporating either a C, an A, a G, or a T, you actually get peaks that are at these predefined sizes. And what you see here in this particular case, and this is an oncology marker actually, you see actually there's a C allele and there's a G allele. So we know this sample is heterozygous. The other thing we also know is the peaks are different heights here. So we know also, because this is an oncology marker, that this particular sample has a cancer fraction of about 30%. In CF, these peaks are normally the same height, because in CF, we only report out two alleles heterozygous normally. There is no fractions there. But in principle, this is how the technology works. We know upfront already what the masses are of the primers, in this case, what the extension products are. And so we can, in real time, define already what the calls are going to be. And then we see other peaks in the spectrum also. These are actually other assays or other alleles from other assays. So here's an example of a group in Ontario that were using the Luminex platform before, and they switched over to our platform. They created a custom panel for CF genotyping, and they actually quoted us as being very flexible and cost-effective alternative to their current genotyping assay. They had a very high concordance with the previous method. They had an increased sensitivity. They found more more mutations because they could get a broader panel. They had a high detection of different variants, and they had a significant cost savings, as you can see here below. On their previous platform, they were spending about $43,000 per number of samples. And after that, when they run these on MassARRAY, they actually only spent $17,000. So they saved about $20,000, $25,000 per batch of samples. Here's the other poster from a group from CDC, and also one of the co-authors actually is Dr. Dhiman. And this was presented at the APHL Newborn Screening Symposium in 2019, the last one before the COVID hit. They actually looked at the CFTR panel that Dr. Dhiman was talking about today, and they actually found that they had a genotyping accuracy of 99.9% when they were testing this with the CDC DPS repository samples. And this is a very nice poster showing that, you know, the panel works really well, and there were different extraction protocols they were using, and they got the same results in all the cases. So what do we have right now for CF at Agena Bioscience? So we have the IPEX Pro CFTR 74 variant panel that Dr. Dhiman talked about, which has the ACOG23 here in blue, plus another 54 mutations. This panel, based on different literature values and the CFTR2 database, is predicted to cover between 75% and 96% of the mutations in different ethnicities. This can be a panel that works really well for you. We have actually a few different large newborn screening sites, public newborn screening sites in America running a panel like this. Or actually, we have some that have designed their own panel that suited their specific needs because they had more preference of certain ethnicities. So what people can do really is we also have a custom design services here at Agena Bioscience called Assays by Agena, where they can actually design custom panels for you that really fit your needs. So like if you have, as I said, a different ethnicity buildup in your area, we can create with you a different panel. You can also say, well, we like say CF74, but we also need a few additional markers. So we could also develop just one multiplex with some additional markers. Or, yeah, you can get a population-specific panel if you are, say, in an area with a high prevalence of Hispanics or Asian ethnicity, then you might want to create a population-specific panel. And that can all be done using our Assays by Agena, our custom design services. So an example of other assays we have developed in the past already for different customers. We have over 200 assays that we have developed in total for CFTR, and these are some of the other ones that were not on the IPEX 74 panel, but that are available. So with that, just a short summary of our advantages to use Agena for CFTR screening. We have like a very broad panel like the CFTR 74 that can be used for quick screening of samples. We can easily create other sets of assays as we have done for other labs already, and we have actually running labs running panels from 43 up to 160 different variants in their panel. And a genotyping assay can definitely be a very cost-effective preliminary screen for like other things like next-gen sequencing, for instance. And as I mentioned, Assays by Agena is a great tool to customize a CFTR panel to your needs if need be. So here is a small picture of our platform, and I just wanted to reiterate here, we are here for targeted actionable results. We want to give you the results you can use now. No variants of unknown significance, really only what you pick. It's very cost effective. It has a very efficient workflow. It's a single day workflow from sample to results. It is customizable, as I showed you already for CFTR, but also if you look on the right side of the picture, we can do oncology, tumor liquid biopsy screening. We have a foothold in pharmacogenetics, other hereditary diseases, and it can also be used for sample integrity. With that, I would like to hand it over to Julie Caro again for a question and answer session.
Yeah, thank you very much, Dr. Evans, and also Dr. Dhiman. for your presentations. If you have a question, please type it into the Q&A panel. And we would also like to ask our attendees to take a brief moment after the webinar has ended to take our exit survey and provide us with your feedback. So with that, let's see what we have for questions. Yeah, here's one for Dr. Dhiman. The question is, are there any old specimens that turned out to be positive but were not picked up with your previous assay? And did you test those with the new Agena assay? And if so, what were the results?
Yeah, so when we were doing our, CLIA validation, when we were investigating this assay, we did use all of our old known specimens that were positive, and they all repeated on the Agena panel. It was 100% concordance. We did not find any older, for any older specimens that say were reported as negative, we didn't find any new mutations. We haven't done something as thorough as that. And a lot of those reasons also include that. Also, the storage, how long we keep these cards. While we keep any positive cards for 6 years at IDPH, any cards that are negative for all disorders are typically only kept for 4 months. So we only have so many specimens, older specimens to test that were negative across the board.
Okay, great. Another question for you. I know you showed us your workflow for screening, but the question is just to make that clear. Is the Agena cystic fibrosis kit used for first-tier screening or for both first and second-tier screening?
Yeah, we use the Agena for the second-tier screening, the mutation analysis. For this the first-tier screen, the IRT, we use a assay kit developed by Perkin Elmer that works pretty well for us.
Okay, great. Here's a question maybe for Dr. Abbott. What are the advantages of the Agena technology compared to known PCR tests that use a genetic analyzer to diagnose cystic fibrosis in newborns?
Well, that's a great question. I think definitely one of the things that stand out for our platform is that you can do multiplexing a lot better. So our technology can multiplex easily 25 to 30 assays per reaction, whereas some of the other technologies, it's individual assays, or you can multiplex up to 4 or 5 maybe. So it is, when you look at the number of wells that you have to run for a sample compared to other technologies, that might be one of the better ways of doing it. It takes less DNA. It can be more cost effective also depending on the technology. So there are definitely several advantages to using Agena versus a straight PCR.
Okay. Another one for Dr. Dhiman regarding the sample preparation. Can you comment on the dry blood spot DNA extraction and any quality control measures you may take prior to amplification?
Yeah, so all cards before they're even punched are visually inspected to ensure that they are properly spotted on the card, as I had mentioned in my talk earlier. As far as prior to amplification, they undergo a standard DNA, pretty straightforward DNA extraction using like the typical QIAGEN buffers. We don't do any like DNA quantification or anything. We just do the amplification and the PCR SAP extension. And then obviously with every single run, we do have positive and negative controls. So in that sense, every run has its own internal QA, QC. So if something is not working and the controls on that specific run, that entire plate is basically rejected, and we basically restart from the beginning with the punch, with the punching and extraction.
Yeah, I think that answers another question we just had that was regarding controls required for each run. You just said you have those, and they were also used for clinical validation when the test was first implemented in the lab?
Yeah, so I mean, any controls that we've used, that we use in our assays in production, are highly vetted during the validation process to ensure everything's working properly in the assay, and repeatable specificity, sensitivity, amplification range, all the typical CLIA validation test areas that are typically used for laboratory developed tests.
Right. Okay. Can you say a little bit about how the Agena assay compares to whatever assay you used previously?
Yeah, so I've only been working with IDPH for about 5 years, a little less than 5 years. The previous assay they were using was a GenMark-based assay by GenMark, which again only screened for the 23 ACMG mutations, and so it worked well. However, again, we were missing a lot of positives, as I noted. Like there were about 100 or so cases every year since we've gone live with this test in March of 2018 that we are encountering, that we're calling out both carriers as well as some few clinical positives that we see with the Agena assay.
Okay. Here's maybe a question for Dr. Everts. So why did Agena decide to go beyond the ACOG recommended 23 mutations, which are supposed to get you 80 to 90% of the cases? What's the reason behind that?
That's an excellent question. So the ACOG, or the ACMG 23 mutation panel, it's really based on a background of mostly Caucasian samples. So within Caucasian as a Jewish sample, you might get 90 to 98% of your cases based on those 23 mutations. However, if you start looking at other ethnicities, such as Hispanics or Asian ethnicities, they actually will only cover maybe 50% of the cases that are found there. So that was one of the reasons that we wanted to have like more a panel that was more multi-ethnic, and we added in markers for other ethnicities to really crank up as much as we could in a certain small 3-well panel.
Okay. Another question just came in regarding sample preparation. Are the controls spotted on paper also, and do they go through the same sample prep process as the real samples?
Oh yeah, so the negative controls are essentially a blank blood spot card. So that's exactly the same specimen. It's just completely blank. So that's just the same. As far as the positive controls, that varies by disorder. Some disorders, we are able to essentially kind of mimic that blood spot punch, where we have like a control that's we spot on the card and then we punch it and it goes through the same extraction process. So for example, for our SCID disorder, SCID screen, severe combined immunodeficiency, we're able to do that. However, for CF, we just don't have controls like that available. So we basically just spike in, or we use a DNA, a purified DNA control that is spiked into the control well prior to application.
Okay. And then a question probably for either of you. Is the Agena assay able to detect a whole CFTR gene deletion or a partial duplication of the gene?
Well, I can take that question. So we have not designed an assay for a whole gene deletion or a but we do have the exon 2, 3 and exon 22, 23 deletion in there. So, but if you know what you're looking for, if you know what the boundaries are, it's easy to design on the platform. That's a nice thing. It's an open platform. So as long as you know what you're looking for, it can be designed on it.
Okay. I'm just looking through a question that just came in. That's for Dr. Dhiman. You said that in 2020, I think you had 6,200 that initially screened positive and went on to the additional screen. That seemed to the participant like a high false positive rate. Can you comment on that? Yeah,
so if you think about the goal of newborn screening is to make sure we try to catch as much as possible, right? It's a screening test. So we're looking at the top 4% of specimens for a given day that fall in the highest IRT. So just because they have a high IRT doesn't necessarily mean they're going to be positive for CF. So it's built into the algorithm that this encompassing you're trying to catch as many high IRT values as possible. And through a lot of these published studies, they've settled on top 3 or 4%. And so those get reflexed to cfDNA. So instead of 155,000, we're only testing 6,200. So yeah, if you're looking at the numbers kind of as this, if you step back and look at the numbers, it seems like a large, rate. But again, we're not calling out those 6,200 as positives, right? Those are just moving to the second tier. And then of those, we then specifically are screening those for cfDNA mutation. And then from those, we are calling out about 500 or so positives per year.
Got it. Okay. This is actually all the time we have for questions today. So let me thank our speakers, Dr. Vineet Dhiman and Dr. Robin Everts, and our sponsor, Agena Bioscience. And if we didn't have time to get to your question today, we will try to have someone follow up with you via email. As a reminder, please look out for the survey after you log out to provide us with your feedback. And if you missed any part of this webinar or would like to listen to it again, we will send you an archived version by email. So with that, thank you very much for attending this genome webinar.